Now the next question is, what are the major biomolecules? Generally, there are 4 major classes of biomolecules, namely protein, nucleic acid, polysaccharide and lipids. Now let us learn each of these major biomolecules one by one. Number 1, proteins. Proteins are long polymers of the monomeric unit called amino acids. Therefore, amino acids are known as building blocks of protein. In protein, each of the amino acids join together and form a long chain by a special type of covalent bond known as peptide bond. It is also known as amide bond. In reality, the protein molecules often contain more than 100 amino acid units. For example, human cytochrome C is a small protein that has 104 amino acid residue. Its protein differ widely in amino acid composition and also their amino acid sequences. Proteins are present in all compartments of the cell and constitute 50% of cell dry weight and hence occupy the largest fraction among the biomacromolecules. Proteins are most versatile. They may act as enzymes and also serve as structural element of a cell, signal receptor, transporters that carry specific substance into or out of the cell. Now let us discuss the general structure and properties of amino acid, the monomer of protein. From the picture, we know that all the amino acids have 2 important functional groups, namely carboxyl group or COOIs and amino group, the NH2 group, and a side chain R group, which is differ in their structure for different amino acids. The amino group and the carboxyl group are highly reactive and involved in the peptide bond formation. However, R group is not involved even the presence of the functional group such as COOIs and NH2 group. There are about 100 amino acids have been shown to present in various plants and animals. However, only 20 of which have the L isomer are found as a constituent of the most proteins. These 20 amino acids are differ only in the structure of their side chain, the R group, and they are often referred to as standard amino acid. And 2 amino acids, namely selenocysteine and pyrrolysine, was newly reported to found as constituent of protein. Selenocysteine is present in several enzymes such as glutathione peroxidase, thioredoxin reductases, glycine reductases, whereas pyrrolysine in some methanogen bacteria. Now, let us learn how the protein is made from their amino acid monomer. Formation of peptide bond is an example of condensation reaction involving losses of water molecule. In this reaction, alpha carboxyl group of one amino acid, say R1, is reacted with the alpha amino group of another amino acid, say R2, and the 2 amino acids are joined together by a covalent bond known as peptide bond. The resulting product is called dipeptide, which contains 2 amino acid units. The peptide made up from 2 to 10 amino acids are called as oligopeptide. The proteins are polypeptide with more than 100 amino acids. The word protein and polypeptide is used as an interchangeable term. The bond between 2 adjacent amino acids in a protein is a special type of amide bond, CONIs, where the carbon atom of COOIs group is linked with nitrogen atom of NH2 group of the adjacent amino acid. The repeated amide nitrogen and alpha carboxyl and carbonyl carbon atoms of each amino acid residue form the backbone of the protein chain. Proteins differ in amino acid composition and their sequences. They may also contain one or more polypeptide and can be linked by disulfide bond in addition to peptide bond. For example, serum albumin, a human protein, is a single polypeptide chain and has 609 amino acid residue, molecular weight of 68,000. Human insulin has 2 peptide chains containing 51 amino acid residue linked by a disulfide bond. Glutamine synthase in E. coli contains 12 polypeptide chains that has 5,628 amino acids with a molecular weight of 6,19,000, and 13, a human protein, is a single polypeptide chain that has 26,926 amino acids with a molecular weight of 29,93,000. Now, structural and biological properties of peptide bond. Peptide or amide bond has a partial double bond character and is always in a trans configuration. They are rigid and lies in the same plane where the side chains are protrude out from the primary chain. Peptide bonds present in a proteins are stable in physiological pIs. Now, N and C terminal amino acid. Each amino acid in a protein or polypeptide chain is termed as residue. The terminal amino acid residue with a free amino group is called N-terminal amino acid, and the one with other free carboxyl group at the other end called C-terminal amino acid. By convention, the N-terminal is taken to be the beginning of polypeptide chain. Thus, in the tetrapeptide tyrosine, glycine, lysine, and alanine, tyrosine is the amino terminal residue and alanine is the carboxyl terminal residue. Now, covalent bonds involved in the protein structure. The principal linkage found in the all protein is the peptide bond or amide bond. In addition to peptide bond, a second type of covalent bond known as disulfide bond is found in the proteins and polypeptides. Disulfide bond join two cysteine residue that present in a primary chain to yield a 1 mole of cysteine. Cysteine is an amino acid with a disulfide base.